Uh, all right. So hello, everyone. Welcome to the second seminar of the INCEPT series. Before introducing our next speaker, Dr. Susana Moles from UMASS Medical School, I just wanted to remind you that there's a couple more events today. At 4, now as you know, at 12:45 there's the seminar, but at 4:30 p.m. we have a more in-depth discussion with the speaker, where we can ask, about her career path or the challenges of the job market she just went through. Everyone is welcome to join using the same Zoom link that we are using now. At 5:30, also we have the HBIOB hour on Mixology. It was organized by Yasmin, a postdoc in the DMECI lab, and one of the INCEPT organizers, and Erkin, a postdoc in the church lab. We will learn about their passion on mixology, and they will show us how to make a couple of cocktails. So take a break, grab a beverage, and join us. I also want to briefly advertise our last seminar by Dr. Steve Del Signore that is happening on March 16. And on that day, we also have organized a career panel on how to navigate the job market during a pandemic. We will have a chance to ask questions that are important for career progression and job search, like if you develop a plan with your mentor, how did you approach virtual chalk talks and stuff like that. Last but not least, I encourage all of you to please take the INCEPT survey. I'm gonna post the link in the chat. We would love to hear from you in order to improve our future INCEPT seminar series. And now I'll hand it over to Charlene. Stop sharing my screen, and she's gonna introduce our speaker.
So I want to welcome everyone to our second INCEPT seminar. We have the great pleasure of having a wonderful woman in science, Dr. Susana Moles, present her work to us today on the International Day of Women and Girls in Science. Susana obtained her bachelor in biotechnology from the Autonomous University of Barcelona with the highest distinction in 2007. And she got her master's in biomedical research from Pompeu Fabra University in Barcelona 1 year later. In 2012, she received her PhD in biomedicine from also Pompeu Fabra University in Barcelona under a competitive JAE predoctoral fellowship in the laboratory of Dr. Mara Diersen at the Center for Genomic Regulation in Barcelona. Her research as a graduate student revealed important aspects of drug addiction in a mouse model of susceptibility to nicotine dependence. From 2012 to 2014, she worked as a research scientist in collaboration with the biotech company Proust Institute for Biomedical Research, where she led multiple projects that addressed the pharmacological effects of candidate drugs to ameliorate cognitive function in neurodegenerative and neurodevelopmental disorders. In 2014, she joined the laboratory of Dr. Andrew R. Taper at the University of Massachusetts Medical School as a postdoctoral research associate. Currently, she has full independence regarding her research, and she is focused on mid-range circuits and how they control behavioral responses to novelty and familiarity. Today, Susana will talk about the temporal and spatial activity dynamics of these circuits. With that, I welcome Susana once again.
First of all, I would like to thank the INCEPT program for inviting me to give a talk today, especially because today is the International Women in Science Day, and I want to send a warm rewards to all the women here at the audience. I want to thank you for this nice introduction, and let me share. Let me share this. Today I'm going to talk about the research line that I've developed while being a postdoctoral researcher at the University of Massachusetts Medical School, trying to understand the circuits that convey novelty and familiarity signaling. Can you hear me well?
Yes.
Yes.
So, the first time that we encounter a new stimuli, whenever we meet someone new, we go to a new place, maybe when we play with new objects or we try new food, our brain engages a high response to these new events. Novelty increases attention, arousal, and cognition, and a high response to novelty is important for evaluating the overall salience of these new unknown stimuli. Are they going to provide me some benefit, or importantly, are they going to be potentially threatening? Guiding an appropriate behavioral response. However, as these novel stimuli become familiar open multiple exposure, the novelty response habituates as a form of behavioral plasticity that acts as a filter for us to concentrate our attention in a more relevant information. The response to novelty and habituation with familiarity are innate responses, evolutionarily conserved among species, and they are fundamental for the adaptation to changes in the environment. They are important for survival. Multiple neurodevelopmental, neuropsychiatric, and neurodegenerative disorders, they manifest disrupted novelty response and habituation with familiarity. For instance, in autism spectrum disorders, there is abnormal response to social but also non-social cues and deficits in habituation. In schizophrenia, there is an overvalue of novelty that has been correlated to the positive symptoms of the disease. Or in drug addiction, there is this high correlation in humans and also in rodents that those individuals that have high reactivity to novelty, they also tend to become more addictive to drugs of abuse. But what are the circuitries and the mechanisms that control a response to novelty and habituation with familiarity? Is it a single circuitry or different circuitries mediate responses to different type of a stimuli? What happens? What's this plasticity when a stimuli become familiar? How are novelty circuitries disrupted in neurological disorders? To what extent this disruption contributes to the disease neuropathology? Importantly, can we manipulate the activity of novelty circuitries to improve or ameliorate some of the symptoms of these disorders? There's a big gap on how the novelty circuits are shaped across lifespan experience. This question is based on the idea that there are different kinds of novelties based on the past experiences. Common novelty refers to this novelty that we have previous representations of or schemas, whereas distinct or absolute novelty refers to unrelated past. They are unrelated to the past. And this experience-dependent plasticity of novelty processing is important to understand the novelty response, not only in health, but also in disease. Today, what I'm going to talk about is on this, what we have learned so far on the circuits and mechanisms that control the novelty response. But as you can imagine, whenever we encounter a new stimulus, our brain engages a wide network of interconnected structures. Among these structures, I like to cite the study from Bunzek, in which this is those were human, that's an fMRI study in human healthy volunteers that were presented to images that were novel, familiar, or very familiar. And what they observe is that exposure to novel stimuli increased the activity of medial temporal lobe structures, for instance, the hippocampus or cortical areas. And there's been an extensive work on how these areas regulate the response to novelty, but more from the cognitive part of recognition. Interestingly, in this same study, they found that exposure to novelty increases the activity of dopamine, midbrain dopamine systems that include the ventral tegmental area and the substantia nigra. The activation of this area is important for evaluating novelty and salience and guiding appropriate approach avoidance behaviors. Novelty increases the activity of these midbrain dopaminergic systems, not only in humans, but also in rodents. Most of you must recognize this work. That's pioneer work on fiber photometry combined with biosensors to provide a real-time measure of neural activity. That's from the Dizer's group. And in this study, what they did was to record the activity of PTA dopaminergic neurons when the animals encountered new social stimuli. And they found that novel social stimuli increased the activity of PTA dopaminergic neurons. Moreover, the group of Dupre, they showed that whenever an animal is exposed to a novel environment, but not a familiar, there is increased firing rate of dopamine neurons in the ventral tegmental area. So novel environments also activate this circuitry. More recently, the group of Oshida here in Harvard, and you might also have seen this work, they have characterized a specific circuitry, dopaminergic circuitry, that originates from the substantia nigra lateral and projects to the most caudal part of the striatum. That's called the tail of the striatum. And this circuitry is specifically activated by when an animal is exposed to a new odor, when the animal is exposed to new tones, or even to new objects. So there is this dedicated midbrain circuitry that responds to novelty, and we know that it looks like there is a topographic segregation of midbrain dopamine ensembles that project different regions, different compartments of the striatum to regulate the novelty response. The BTA dopaminergic projections to the nucleus accumbens is important for novel social, but also the BTA has been activated by environments and objects. And there is this specific circuitry that responds from the substantia nigra to the tail of the striatum that responds to new odors, that it goes novelty and physical salience. So there is this dedicated circuitry for novelty, and the question is, what happens when stimulus becomes familiar? Is it the same circuitries, or other circuits might also be involved? My research has been focused on a very unique circuitry of the brain that's called the medial abenula interpeduncular axis. This is evolutionary conserved from fish to mammals. The medial abenula is an epithalamic structure that in a sagittal view of a rodent brain, you can see that it sits just below the hippocampus. The medial abenula receives cognitive and sensory information from the septum and conveys this information almost exclusively to the interpeduncular nucleus of the brain, or IPN, which sits next to the ventral tegmental area. So as a part of the abenula complex, this axis acts as a bridge, conveying forebrain cognitive information and linking this information to midbrain regions connected to emotional and motivational behaviors. So the way the medial abenula projects to the IPN is very unique, and the ventral part sends cholinergic, glutamatergic inputs that innervate the most part of the IPN, and that provides excitatory input to the IPN. Instead, the more dorsal part of the medial abenula sends substance P signals that mostly innervate the lateral part of the interpeduncular nucleus. This axis was kind of unknown, and it was the work from Paul's Kenny laboratory that they discovered that the medial abenula is important for controlling aversion to nicotine intake, where people start to become interested in this circuitry. Our lab and other research have shown that the medial abenula interpeduncular axis is involved in nicotine withdrawal and controls the addictive properties of several drugs of abuse. Moreover, our other investigations have linked the medial abenula IPN with emotional behaviors of anxiety and fear responses. So given that novelty uses the same circuitries of drugs of abuse, and this medial abenula IPN axis is also involved in drug addiction, we started a project to ask whether this axis might be important for the response to novelty. And this work was published a few years ago, in which we demonstrated that this circuitry is important for familiarity signaling and for responses of novelty preference. And here, basically what we did was we took wild-type animals, and those were presented to new social stimuli that became familiar upon multiple exposures. That's the familiar social group. Or we had another group of animals that this familiar social was replaced by a new one. What typically happens is that on the first day, there is increased exploration of this new social stimuli that decreases as it becomes familiar. However, by placing new social stimulus on the third day, this novelty drives a rebound investigation. And in these animals, we measured the activation of the IPN with CFOS. And to our surprise, what we found was that the number of CFOS positive nuclei in the IPN were much higher in the familiar social group compared to the novel. And these CFOS colloquialize with GAT1-2, that's a marker of inhibitory neurons, suggesting that in the familiar social group, there is activation of IPN converging neurons. We found similar results when animals were presented to other types of familiar stimuli, such as new familiar inanimate objects. So in another group of animals, they were presented for consecutive times to a new inanimate object that became familiar. That's the familiar object group. Or another group of animals that were presented to a new object. We observe increases in investigation on the first day that were reduced as this object became familiar. And if we placed a new object, that caused a rebound investigation. When we measured the activity of the IPN in these two groups, we observed more CFOS expression in the familiar object group as compared to the novel. So all these results led us to the hypothesis that when a stimulus becomes familiar upon multiple exposures, this activates IPN GABAergic neurons. The IPN is mainly composed of inhibitory neurons. It's an inhibitory nucleus in the brain. And this activation of the IPN acts as a brake of novelty-induced exploratory behavior. So we wanted to test this hypothesis, and for that we used optogenetics to selectively activate or silence the activity of IPN GABAergic neurons. So we injected Cre-dependent helorhodopsin in the IPN of GAT2 pre-animals or channel rhodopsin, and then we start silencing the activity of these IPN GABAergic neurons when the animal were presented to a familiar or a novel social stimuli. What we observe is that silencing the activity of IPN GABAergic neurons caused an animal to increase the familiar social investigation. So the animals treated the familiar as if it would be novel without disrupting the novelty social. And this affected the novelty preference ratio. Conversely, when we activated with channel rhodopsin these IPN GABAergic neurons, we found a reduction in novel social investigation. So the animals treated the novel as it would be familiar, and that disrupted this novelty ratio. So the activity of IPN GABAergic neurons was necessary and sufficient for the expression of familiarity. We observe similar results when the animals were presented the choice between a novel and a familiar object. So when we silence the activity of these neurons during this choice paradigm, we observe increased investigation of the familiar object and disruptions in the novelty preference ratio. Conversely, activation of these GABAergic neurons decreased novelty investigation, and that affected the novelty preference ratio. we wanted to go a little bit farther, and we did a little bit of circuit analysis, and we investigated whether the medial venula, which is upstream the IPN, might be involved in these familiarity responses. And actually that was the case. So for these experiments, what we did was to inject the Hiller rhodopsin or the China rhodopsin in the medial venula of chat-free animals. This is the major excitatory input in the interpeduncular nucleus. And then we place an optic fiber in the peduncular nucleus to either silence or activate these axons. And we did observe that when we remove medial venula input, the animals increased the familiar social investigation, and that disrupted the novelty preference ratio. Conversely, when we activated these axons with channelrhodopsin, the animals reduced the novel social investigation. There was this familiarity signaling affecting the preference ratio. A novelty response is triggered by activation of midbrain circuitries, and actually the IPN sits just next to the ventral tegmental area. Is there any direct connection between the ventral tegmental area and the IPN? And actually this is the case. So if you inject the Cre-dependent virus in the ventral tegmental area of that Cre animals, you can see these axons, dopaminergic axons, innervating the interpeduncular nucleus. And Stephen de Groot, he's a former PhD student in the lab. He did a lot of slice physiology, and he determined that those axons are functional. They provide dopamine signaling in the IPN, which is blocked by a D1 receptor antagonist. We wanted to know whether these dopaminergic axons were involved in the response to novelty preference. So we stimulated these axons when the animals were presented to a familiar and a novel social stimuli. And we did observe that the stimulation of dopamine signaling in the IPN drives a novelty signaling. The animals treat the familiar social as if it would be novel, disrupting the novelty preference ratio. Moreover, in this study, given that the ventral tegmental area sends dopamine inputs in the IPN, we wonder whether there are, and based on Stephen's findings, they were blocked by D1 receptors. We wondered whether there are some neurons in the IPN that express the D1 receptor. So to address this question, we use a D1 reporter T-tomato line, and we observe dopaminoceptive neurons in the interpeduncular nucleus that express D1 receptor, and those are located more towards the caudal part of the IPN. There is a rostrocaudal gradient of D1 expressing neurons. We use these D1 tomato animals to see whether these neurons show any kind of response to novelty. So these D1 T-tomato animals were presented to a familiar social stimuli or a novel social stimuli using the paradigm described earlier. Oh, can you hear me?
Yeah.
Sorry.
You're muted just for 1 second.
Okay, good. So what we did observe with this experiment is that the interaction with the familiar social increases the IPN activity. However, in those animals presented to a new social stimuli, there is increased activation of D1 positive neurons. So some neurons in the IPN respond to novelty, and those are the ones that receive that express D1 receptor. So we wanted to know whether the activation of this D1 is important for the novelty signal that we drive by photostimulating the axons coming from the ventral tegmental area. So we pre-infuse D1 antagonists before photostimulating dopaminergic axons in the IPN, and we were able to completely rescue this impairment in novelty response that we observe by promoting dopaminergic signaling. So we found that that novel stimuli activate the ventral tegmental area, and specific subpopulations of the IPN that express D1 receptor. However, as this new social and object become familiar, this familiarity activates interpedagogic neurons in the interpedunculaire nucleus through the medial venula, which act as a brake of novelty-induced exploratory behavior. And that was a new circuitry responding to within the novelty response. So the medial venula IPN integrates into predefined circuitries that we know that have been reported in the literature to respond to new social and new inanimate objects. So now we have characterized this new circuitry, and we want to what we want to know is to define specific patterns of activity dynamics that might correlate with these behavioral responses of novelty and familiarity. Moreover, we want to identify whether there are specific circuitries within the IPN showing these patterns of activity that might convey this information and are involved in novelty preference responses. So most of my data that was published use CFOS as a marker of activation, but the CFOS doesn't provide a temporal readout of what's the activity dynamics of these circuitries during novelty and familiarity responses. In the lab, we have established fiber photometry combined with biosensor to measure in real time the activity dynamics of these circuitries, which we believe that this dynamic is fundamental for the novelty response. And for example, we know that the ventral tegmental area dopaminergic system responds to novelty, but not to familiarity. But is there any activity trace, any signature in these activity dynamics that could reflect the transition between novelty to familiarity? Moreover, there are no reports on how these novelty circuitries are engaged when an animal has a choice between a familiar and a novel. To answer all these questions, we have started by recording dopaminergic activity in the ventral tegmental area. So we have injected GCAM, cre-dependent GCAM, in the VTA of DAT-CRE animals, and we have recorded activity of these neurons when the animals are presented to new social stimuli and when they become familiar, also when there is a choice. So what we have observed, this is on the first day of encountering a new social stimuli. We see patterns of activation of these dopaminergic neurons. We have time-locked these activity patterns based on the animal behavior, in particular when the animal investigates the social and non-social cylinders. And whenever the animal, we consider onset of exploration whenever the animal approaches and starts an investigation of these cylinders. So what we have found is that these increases in activity of dopaminergic, that this increased activity of dopamine neurons, they coincides with when the animal investigates the social cylinder, suggesting that sociability increases the activity of BTA dopaminergic neurons, and that trace is very similar to what was previously reported in Deiser's group, although this was done when there was direct contact between these two animals. And what we have in common between these two studies is that the activation of ventral tegmental area dopaminergic neurons is always higher on the very first bout of interaction. That's what they observe, and that's what we observe in our recordings. And that indicates that BTA activity not only encodes sociability, but it encodes the novelty component of social interactions. So on the second day, when now this social stimuli becomes familiar, we see this activity pattern. We again detect increases in dopamine activity when there is investigation of the social cylinder. And when we pay attention to this very first bout of interaction, we see increases of activity, but are of lower magnitude. So there is an habituation of how PTA dopaminergic neurons respond. There is this habituation of PTA dopaminergic activity as the social stimulus becomes familiar. And this habituation is even more pronounced on the third day that this social stimulus is more familiar. So we have quantified these responses, and we can observe very clearly that there is a decreased response with social familiarity. But there's something that I would like to highlight here, and that strikes us, is that on the first day, when we pay attention to this first interaction, the raises in dopaminergic activity in the PTA, this raising activity occurs after the animal starts this social investigation and reaches the higher level after 4 to 5 seconds once the investigation has begun. However, on the day 2 and 3, when the animal is familiar, the raise of PTA dopamine activity began much earlier, before the onset of exploratory event. So we think that this shift in activity that represents a trace, an activity signature that represents behavioral forms of social learning, and that might be involved with familiarity. So these results are telling us that PTA dopamine activity underlies somehow social predictability. And it might be possible that different circuitries engaged in the PTA are different from day 1 to day 2, from the first day as compared to the others. It's been well established that the ventral tegmental area dopaminergic circuits to the nucleus accumbens mediate sociability. So we wondered, what is the real-time dopamine release in the nucleus accumbens during this sociability test when the animals encounter a new social stimuli and when this becomes familiar? So we have measured dopamine release, in vivo dopamine release, using the D-Light dopamine biosensor injected in the nucleus accumbens of wild-type animals. And we have been able to detect specific patterns of dopamine release across the different days. Nicely, these patterns resemble the ones that we observe when we record BTA cell bodies. And we have, again, time-locked these activity patterns to behavioral events of exploration of the social and non-social cylinder. And we observe very similar responses as in the BTA. So we see a high activation, a high dopamine release in the nucleus accumbens the first day when the animal investigates the social cylinder. So sociability increases dopamine release. And this dopamine release reduces as the animal becomes familiar. But interestingly, again, this release of dopamine on the first day occurs after the onset of exploration. However, on day 2 and day 3, there is this raise in dopamine occurs much earlier, suggesting that this nucleus accumbens dopamine release also might underlie social predictability. Going back to the ventral tegmental area, we wanted to know what is the activity of these neurons when the animals are offered a choice between a familiar and a novel. Here we observe again increases in activity that coincide mainly when the animals explore the novel and as compared to the familiar. So the PTA dopamine activity encodes social novelty in this task. But as you can see here, we do observe that this raising activity when the animals approach and investigate the non-social also began before the onset of investigation. So this PTA dopamine activity during a choice, in the choice paradigm, responds to novelty, to social novelty, but in a context-dependent manner. We have been also interested in knowing how is the activity of IPN GABAergic neurons during this choice paradigm. So we have injected the CRE-dependent GCAM in the IPN of GAD2 CRE animals and place a fiber to record from these neurons. And nicely, what we observe is the opposite patterns as we have seen in the ventral tegmental area. So whenever the animal approaches and investigates the familiar social stimulus, there is increased activity of IPN GABAergic neurons. However, whenever the animal approaches the new social, this coincides with reductions in neuronal activity. And considering this, that these changes in activity occur before the onset of investigation, we think that this, the IPN GABAergic activity might somehow encode this predictability of social encounters in an opposite way if in social, familiar or novel social investigations. So a huge part of this project has been devoted to understand where the IPN conveys information. The IPN is connected to many brain structures, and there are few investigations on the functional role of these connections. If we go to the Adlan Brain Atlas and we observe patterns of connectivity, IPN inhibitory neurons, they establish these inhibitory networks. But if you pay attention, this is work from Turner's laboratory in a NOS-CRE animal. Most of the IPN GABAergic neurons, they send inhibitory projections to the most caudal part of the brain, the hip brain. What about our GAD2 CRE our GAD neurons? Are those interneurons or they are projecting neurons? When we inject a CRE-dependent EGFP in the IPN of GAD2 CRE animals, we do observe very similar pattern of projections. These IPN GAD2 neurons send axons to the median raphe and also the dorsal raphe. And they innervate regions such as the nucleus incertus or the lateral dorsal tegmental nucleus. We are particularly interested in the LDTG because the LDTG provides excitatory input to the ventral tegmental area dopaminergic neurons. So if these IPN GAD2 neurons send inhibitory tone to the LDTG, that might represent like a feedback mechanism to the novelty circuitry. So this LDTG might be a good candidate for responses of novelty. Actually, there is work from the McGill laboratory that they have identified some circuitries between the IPN, inhibitory circuitries between the IPN and the LDTG, and those are involved in nicotine aversion. So the first thing that we have done is to confirm that there is a direct connection between the IPN and the LDTG. And for this experiment, we have injected retro-LOX EGFP in the LDTG together with wild-type and cherry virus to know the injection site. And when we go to the interpeduncular nucleus to see which neurons project in this region, we observe a rostrocaudal gradient again. We are unable to see cell bodies in the more rostral part of the IPN, but we can detect neurons that project to the LDTG area in the more caudal and ventral part of the interpeduncular nucleus. And like a tricky experiment of this project has been trying to record the activity of this subcircuitry of the IPN that projects to the LDTG. And for these experiments, what we have done is inject retro GCAM in the LDTG of GAD2 CRE animals, and we placed an optic fiber in the IPN. And we have measured different responses. I'm going to present here what we do observe when the animals are offered the choice between a novel and a familiar social stimuli. And we do observe activity patterns that resemble when we record from the whole IPN. whenever the animals approach the novel social investigation, this coincides with reductions in activity of this subcircuitry, that these reductions are less relevant for familiar social investigations. So this IPN to the LDTG circuitry seems to be inhibited for an animal to explore new social stimuli. So we hypothesize that if we activate this subcircuitry, we might affect the novelty exploration, and that's exactly what happens. What we have done is inject retro control virus or retro channel rhodopsin in the LDTG of GAT2 animals, and we photostimulated this subcircuitry by placing the fiber in the IPN when the animals are presented to familiar and novel social stimuli. Here we have applied more precise closed-loop stimulation protocol, and what we do observe is that activating these IPN LDTG neurons reduces the novel social investigation, and that disrupts the preference radio. So this circuitry is sufficient to inhibit novelty exploration. We have seen these effects not only with new social, but also with new objects. So in my working model, where we suggest that activation of IPN converging neurons inhibits novelty-induced exploratory behavior, this is through projection, specific projections through the LDTG nucleus. Okay, I will just finish presenting some ongoing and future research projects that I'm working with. A major goal of my research is to provide a deep understanding of the circuits and mechanisms behind novelty processing. For a long time, I've been studying this response to novelty by investigating how animals react to new social and new object stimuli. These two types of stimuli, the social, we know that it has a positive valence and the object a neutral valence. But for these kind of responses, what is important is that the animal has to approach this stimuli to generate a novelty response, a novelty-induced exploratory behavior. So more recently, we have wondered what happens when the animals are presented to new stimuli that they have negative valence and that they engage avoidance behaviors, for example, a dark shadow. We have preliminary data that this kind of negative stimuli, they inhibit the activity of BTA dopaminergic neurons because the animal doesn't want to engage an exploratory response, but the other way around. And this kind of new stimuli, they significantly activate the IPN GABAergic neurons. So for a complete understanding of these novelty responses, we are including different type of, we are including new stimuli, but they with different valence. And currently we're also working on which kind of neural subtype in the IPN are involved in this novelty response and mediating different types of response depending on the valence of this new stimuli. So the GABAergic neurons in the IPN, they are very diverse and they cluster into specific subnuclei of the IPN, and providing this deep understanding, it's going to help us to completely define the network, how the IPN controls novelty responses. Besides all this circuitry analysis, an important goal of my research is to understand or to address what's the translational perspective of all this work and investigate novelty circuitries in neurological disorders. At the beginning of the talk, I said that in autism spectrum disorder, there is a normal response to novelty and habituation with familiarity. In autism, there are two core symptoms. there's impairments in social communication and interaction, and the manifestation of repetitive behaviors. Those core symptoms are often comorbid with behavioral and cognitive abnormalities. The prominent feature of in autism is this habituation deficit that has been correlated to more severe symptoms of the disease. And moreover, this lack of habituation has been related to the core social but also non-social symptoms of autism. So I believe that if we can understand the circuitries behind these habituation deficits and how they are disrupted in autism, that is going to be of clinical relevance for designing new approaches for the disease. There's been an attempt to investigate which neuropathological circuits are altered in autism by using different mouse models of the disease. And you can see that the prefrontal cortex, the striatum, those are significantly altered in several disease models. I want to highlight that the mental tegmental area, which is a key element in this novelty response, is also has been shown to be disrupted in several models of the disorder. This is work from Camilla Bellone and also Peter Schaeffel, that they are in Switzerland, and they have found that abnormal dopaminergic signaling in the Chang3 or Lignin-3 models of autism might underlie some of the disease features. So as a couple of years ago, I started a project to investigate how the novel circuitries, and in particular the IPN, might be disrupted in autism spectrum disorders. And I chose the Fragile X syndrome. So Fragile X syndrome is the leading monogenetic cause of autism. About 40% of male and 20% of female individuals with Fragile X meet the criteria for autism. It's also the most common inherited form of intellectual disability. It's caused by an expansion and hypermethylation of CGG repeats in the 5' untranslated region of the FMR1 gene, and this causes a loss of FMRP protein expression. So the FMRP protein is a translational repression, and without this protein, there are numerous morphological and synaptic alterations. And as I said, there's an abnormal response to new social stimuli in Fragile X and habituation deficit that have been shown to correlate with the levels of FMRP expression. The FMR1 knockout that recapitulates some of the symptoms of Fragile X. Interestingly, this animal model has altered protein synthesis, especially in the middle of penula, which is just upstream to the IPN. So that led us to hypothesize that this circuitry could be impaired in Fragile X. And in order to investigate whether this IPN might be disrupted in this mouse model of Fragile X, I asked George Richter, who is an investigator here at the UMASS, if he could share some animals for preliminary experiments. And I took some wild type and FMR1 knockout to investigate sociability and activation of the IPN. And actually what I found is that the FMR1 knockout doesn't show this familiarity signaling and shows an impairment in novelty preference ratio that has been described in many other studies. And interestingly, this disruption of novelty preference correlates with abnormal or reduced activation of the interpeduncular nucleus. So I'm happy to say that I've been granted a project from International Brain Research Organization to use more genetic strategies to identify signature tracings of neural activity in the FMR1 knockout and see how these abnormal activity dynamics in novelty circuitries might reflect this sociability and novelty impairment, and also this disrupted habituation, and whether if we can manipulate these circuitries, we can rescue some of the alterations in this disease model. Here, I'll briefly summarize what I've been presented in this last part. That's more unpublished data. We have identified that the mental tegmental area dopamine activity dynamics, and in particular dopamine release in the nucleus accumbens, encode a social novelty and also social predictability. In a choice paradigm, we have identified that BTA dopamine activity encodes a social novelty, but in a context-dependent manner. Moreover, we have defined activity dynamics of IPN GAD2 neurons, and particularly projection to the LDTG, that they show the specific responses to social novelty and familiarity. And this circuitry is important for responses of novelty preference. Finally, there I have preliminary data that might indicate the IPN might be involved in Fragile X, and studying new circuitries that could underlie some of these models, it would be relevant for designing new therapeutic strategies. With that, I want to thank Andrew Tupper, who has been my supervisor, and he has been very supportive along my career. Also Rubin, that she has helped me a lot with many experiments. The founding sources, and I'd like to thank you for your attention. And I'll be happy to take questions, comments that we can address now, or you can send me an email and I'll be happy to reply. Thank you.